And the next speaker is Dr. Bill Andrews. He's a pioneer, has a company called Sierra Sciences. He's the CEO of the company, and they're focused on telomere-based therapies. You guys know what telomeres are? So we got our cells. Our body is made of cells. The cells have the genome. And the genome, on the tips of the genes, have these things called telomeres. And Dr. Bill Andrews is a pioneer on finding and discovering new therapies and technologies to ensure that these telomeres are kept as long as possible in order to keep us as healthy and as young as possible. This is a very short summary. Dr. Andrews, thank you so much, as always, for joining us. It's great to have you. The stage is yours now.
Okay, well, thank you very much. There's a barrier to the amount of lifespan that we can have. And it's something that is very important to me and all of us to try to figure this out. Now, I've got a strong background in statistical theory, and I'm very well aware of the fact that if aging was something that was totally due to random occurrences, such as environmental impacts and stuff like that, the bell curve for how old we would be when we died would be a much broader curve than it is. We would be seeing some people dying of old age at 10 years old, and we'd be seeing some people die of old age at 1,000 years old. But that's not the case. We see a very, very narrow bell curve peak that is suggesting that there's some kind of barrier that we have to solve. And I'm very interested in solving that. As you'll hear, I've been thinking about this my entire life. Now, I'm on the board of directors of the Coalition of Radical Life Extension because I think they're the most enthusiastic group. I don't know why a slide just showed up on my screen here because I didn't operate it. Yeah, it's like, okay, so I think, yeah, so now I've got back to control here. Okay, so the Coalition of Radical Life Extension is the most enthusiastic group I've ever met in terms of trying to figure out how to get us to exceed this barrier of super longevity. And I highly recommend that people join this. My interest in actual aging or the limit on our longevity began when I was really young. You know, my parents sent me, or my teachers sent me home with a note on my shirt to my parents saying, your son's very interested in science and medicine. You should nurture this. So my father came out to the front lawn one time when I was actually looking in through a telescope at Jupiter and Saturn and stuff like that. And he said, Bill, since you're so interested in science and medicine, when you grow up, you should become a doctor and find a cure for aging. He actually used that expression, cure for aging, and that's stuck with me ever since. I mean, a lot of people don't like it, but that's the term I use, curing aging. And he also said, which was kind of interesting, he said he didn't understand why nobody's done that yet. He thought it would be something simple. I thought, okay, maybe it is simple. And so, but I started realizing, you know, I really love living. And I would hate to have living become any less lovable for me or anyone just because of this thing called aging, okay, this limit on our lifespan. And so I just decided to really take this on. And all through college and high school and college, I was known as Mr. Cure Aging. I would start clubs. I'd have a lot of meetings. We'd be just discussing the theoretical aspects of why we age, how we age, all these kind of things. Now, all the theories seem to be based on environmental impacts, and all the twos and twos didn't add up to me, okay? Because as I said, the bell curve was too narrow to be explained by environmental impacts. There had to be something else. I decided there had to be some kind of clock that's inside of us. Then one day, one of my friends at one of our meetings mentioned that, you know, some guy named Leonard Hayflick had discovered that human cells can only divide a certain number of times, and then they stop. They go into a phase called senescence. And he said, nobody believes him. Everybody thinks he's made some big mistake, some artifact in his studies. But I thought, oh my God, he's right. That's it. That's got to be it. This is that kind of clock. It's a clock. It's not a clock of time. It's a clock of events. But fortunately, there's a lot of cells in our body that divide at a constant rate, and so it is essentially a clock of event that is also a clock of time. And this could be the clock that I'm trying to think of. But then the question became, and we would discuss this in detail, how could a cell possibly know how many times it has divided and how many times it has yet to divide? What kind of I mean, we can imagine a computer or something like that, if we can invent something that could be a clock like that, but how would a biological clock get evolved to do something like this? And this used to really bother me. And then one day I was playing pool in Palm Springs, California, and I knocked a ball into the hole, and I instantly took my cue stick and I put it up against the wall. And I mean, I don't know how they keep track in pool nowadays, but you take your cue stick up and you move this little wooden bead from one side of the string to the other to mark that you've knocked a ball. And I remember thinking, oh my God, I wonder if there could be some biological mechanism like that that is explaining how a cell would know how many times it has divided and how many times it has yet to divide. Well, then I started realizing, a cell doesn't really need to keep track of how many times it has divided. And so I thought, we don't need to move that bead from one side to the other. We just need to remove that bead. Well, that then got me to thinking, hey, that's a lot like ride tickets at an amusement park. Okay, your mother sends you to an amusement park, says, hey, go ride the rides. She gives you 10 tickets. You ride one ride and you only have 9 tickets left. So you have 9 more rides. Then you go on another ride, you have 8 tickets left. So you have 8 more rides. So I thought, wow, is there some kind of mechanism like this that's inside of our cells? But, at the same time, I was asking, why would we evolve something so devastating? Why did we evolve a cure for aging? I've got to turn my phone off here because it's ringing. Sorry. So I went and got a PhD in molecular and population genetics. Population genetics is pretty much the how and why of evolution, instead of like the what and when. Most of us, when we talk about evolution, we talk about what happened and when. But I wanted to learn about how and why. And the molecular part of my degree was also, well, what are we going to do about it? Molecular genetics was how to deal with any kind of evolutionary problems. Humans are smart enough to control their own evolution now, and molecular genetics is the way to do it. So during this process, I began to understand why we evolved an aging process, which would be too long to explain here. But I also learned the tools to how to fix it. But I still had no idea what those ride tickets were. And so when I got my PhD, I went into biotech and worked on cancer and heart disease and inflammation. And I had actually a very successful career doing that. I was involved in a lot of the big biotech breakthroughs. But I kept thinking to myself, we're all going to die if we don't do something about this barrier of super longevity. Well, then 12 years later, I was sitting in a conference and I heard a guy named Cal Harley talk about telomeres. Telomeres are found at the tips of our chromosomes, and he pointed out that they get shorter as we age. They get shorter as our cells divide, okay? And he said this is probably the explanation for Leonard Hayflick's discovery about why cells can only divide a certain number of times. But we're conceived when our telomeres are 15,000 bases in length. Bases are units of measurements of DNA. But by the time we're born, our telomeres have already shortened down to 10,000, as I've shown here in this slide already. But it doesn't stop there because we still have a lot of growing to do, a lot of cell divisions, a lot of diseases to fight, a lot of wounds to heal. And when our telomeres get down to 5,000 bases, our cells lose the ability to function and we die of old age. These are the ride tickets that I've been looking for. And it's an actual ticking clock that we have inside of our cells. It's the only one I've ever seen so far or heard of that even makes any sense. But this is something I decided that I'm going to focus on. But, because of this ticking, our cells can only divide 50 times. Our lifespan is limited to 125 years. That's the barrier. We have this barrier of 125 years that we cannot exceed because of telomere shortening. Now, none of us actually live to be 125 years because none of us have the perfect genetics or lead the perfect lifestyle, and things can happen to accelerate that. But when the telomeres get shorter, our gene expression patterns also change. Our young cells start behaving like old cells, even in the petri dish, and our cells become senescent. This is the main cause of senescence in humans, telomere shortening. And Calvin didn't just talk about aging. He talked about a lot of things. And he pointed out that there was a common theme. The common theme is that there's cell division, and when there's cell division, telomeres get shorter, and eventually telomeres get too short, and then cells can no longer divide, and we die. That's the common thing. But the other things, he talked about, like when you drink alcohol, alcohol kills cells, especially liver cells. And then that causes this common theme to occur, except now the cell division is increased. Okay, so we have accelerated cell division. The telomeres still get shorter, but now they get shorter faster. The telomeres get too short, but they get too short quicker. And then cells can no longer divide sooner. And we get liver cirrhosis. That is a major cause of liver cirrhosis, is from drinking all the alcohol, killing the cells. The cells that early on can divide, other cells can divide to replace those cells, but when the telomeres get too short, that can't happen and you get liver cirrhosis. Well, it's just not the alcohol. It's, when you have a muscle cell mutation that causes muscle cells to die quicker, that can result in muscular dystrophy. When you microdermabrasion to treat your skin, to make your skin younger, well, actually it's making it older because the telomeres are getting shorter and you get old feeling skin. HIV kills immune cells. Other immune cells divide to replace them. But that's why AIDS doesn't show up until long after you got infected. And that's AIDS is another thing that's caused by shortening telomeres. Any kind of toxin that you put in your body that's going to kill cells is going to cause accelerated telomere shortening because other cells are going to have to divide to replace those cells, and we die sooner. This is not a theory. This is, as I say, aging might be a theory, but the limit on our lifespan being controlled by telomere shortening is not a theory. Anybody in the world that works on human cells grown in a petri dish knows that when you culture your cells in a petri dish, they can only divide a certain number of times because the telomeres get shorter and the cells senesce. Telomere shortening limits our lifespan just like it limits the cell divisions in the petri dish. Now, this might be a cause of aging. I can't say it is, but I guarantee it's a limit on our lifespan. But we won't know if it's a cause of aging until we actually figure out how to prevent telomere shortening or re-lengthen telomeres. And what's more important is it might actually be the thing that controls all of the theoretical causes of aging. Okay, everything might actually come under control of simply just a telomere. That's a possibility, but we won't know until we lengthen telomeres and see what happens. But the bottom line, no matter what else we do to try to cure aging, nothing else will ever cure aging unless we also solve the telomere shortening problem. So this is an absolute thing we have to do, and this is where all of my energy is going to right now. All right, so what are we going to do about this? I started my own company to work on this problem. I founded the company in 1998. The company is called Sierra Sciences. We're still around. Our one and only mission is to figure out how to overcome this obstacle, how to add more ride tickets back to our telomeres. And the first clue came with the fact that, I said that when our cells divide, our telomeres get shorter, but that's not true in all of our cells. Okay, it turns out in our reproductive cells, they don't get shorter. And this is important because if they did, our children would be born with shorter telomeres than we have. And their children would be born with shorter telomeres than they have, and we would have been extinct as a species millions of years ago, okay, because we wouldn't have any DNA left. So our reproductive cells have figured out some way to prevent telomere shortening. So before I started Sierra Sciences, while I was still at Geron Corporation working with Calvin Harley, because after I heard about his talk, I instantly jumped ship and went and worked with him. I led the team that discovered human telomerase. And so this is a picture. The green squiggly thing is the DNA molecule. Telomerase is this factory-looking thing that binds to the end of the telomeres and re-lengthens them by adding new bases to the telomeres. Okay, so now Geron, they were more interested in the cancer implications. By inhibiting this enzyme, you could essentially kill cancer cells. There's some problems with that that's not working, but they were less interested in aging. And so I started Sierra Sciences to work on promoting the aging, even though I'd already been awarded National Inventor of the Year for my cancer research. So I have a lot of drugs that have been in clinical studies. I'm the inventor of a meltastat that is in clinical studies right now for treating cancer. My mission was to cure aging, and I believe cancer is a result of aging. One of the main causes of cancer is aging. So the research that we're doing is we're trying to develop nutraceuticals and pharmaceuticals that will actually get inside your cells and produce telomerase, just like you have telomerase in your reproductive cells. That means the gene is there. Shouldn't we be able to produce telomerase in all of our cells? So nutraceuticals and pharmaceuticals is something that is a primary mission of Sierra Sciences. What's nice about these is that it'd be a very inexpensive pill you can just swallow. It gets in your blood. It gets to all the cells of your body and lengthens your telomeres. Unfortunately, we've got a lot of research yet to do because we don't have something that produces as much telomerase as we find in our reproductive cells yet. And I'm going to come back to that. But another thing that we're working on, which is really great, is gene therapies. Now, gene therapies are producing enough telomerase to prevent telomere shortening and actually lengthen our telomeres. But the problem is that they are too expensive to really take on. But Liz Parrish will be talking about gene therapies tomorrow. I definitely recommend you listen to that. But imagine, even though they're really expensive, imagine injecting a gene therapy into the discs of the spine. That's a very small area. You wouldn't have to use very much gene therapy to do this. To inject it in the disc of the spine to prevent degenerate disc disease. Prevent and possibly reverse. In fact, really reverse because it's already been done in dogs. Dogs, very beautiful data that's been published in peer-reviewed journals showing that inducing telomerase expression in the discs of dog cells actually prevented and reversed degenerate disc disease. That's not going to be very expensive. We're just looking for a partner that wants to take this on because, let me make it clear, Sierra Sciences is just a research company. Almost every drug that's ever been on the market was never invented by the company that markets it. It was invented by some other company like mine that then licensed it to those companies. So we're looking for partners to take this on. Now, another thing that we're doing is diagnostics. I said before that one of the main causes of cancer is aging. So we're actually doing a lot of work on cancer. Okay, so we're trying to use telomerase activity and stuff like that to identify cancers. We think it's the best cancer diagnostic ever. Again, we're looking for partners. But we're also trying to improve on telomere length measurement protocols because a lot of them exist out there. You can go get your telomeres length measured. I think they have a lot to be desired still. They're not consistent enough, but we're trying to develop newer techniques for doing that. Now, people, partners and stuff like that, they could be taking our discoveries and turning them into supplements, skincare products, pet care products, even stem cells. Everybody working in the stem cell field knows that, especially when you take stem cells from an older person, you can't get them to divide very much because of telomere shortening. So providing a telomerase inducer to stem cells is something that's a very big market for. And we're looking for partners to take this on. But in our efforts to try to find a drug or nutraceutical, so pharmaceutical or nutraceutical, that will cause telomerase to turn on, we've already screened 350,000 chemicals in our high throughput robotic assay. We have a very proprietary assay for detecting telomerase in cells, and we can test up to 4,000 chemicals a day using our system. In that, we have found 900 telomerase inducers, just none of them induce telomerase to the level that we already see in our reproductive cells. Our very best one, though, is produces about 16% as much of the telomerase is seen in our reproductive cells. But the good news is, at least that's slowing the rate of telomere shortening. Now imagine taking this chemical that is 16% produces 16% of telomerase, putting it in something like a salt shaker, and using it as a food topper on your food or your pet's food. There's already all kinds of pet food toppers, but imagine putting this on a human's food topper too. This would be a really great thing. And I think, believe it or not, I believe that this will extend that limit on our lifespan by at least 16%. But our goal is to get to greater than 100%. We're still working there. We still have a ways to go. And so my vision, excuse me for a second. My vision is I want to change the world. Okay, just like I'll move some stuff here. I want to change the world. I mean, it's a very big mountain to climb, but you know, I want to see the accumulation of knowledge in a human brain never cease. I mean, look at David Attenborough. He's 96 years old. He's the guy that wants to rewild the world. I mean, every time I listen to the guy talk, I'm just in awe of the brilliance in that mind. Would it be awful to lose him? Let's not lose him. Let's figure out a way to keep him from reaching that barrier, which he's closer to than most of us right now. Let's get that done. I also want to see us not lose a decline in our bodies as our fitness decline. And the loss of Jack LaLanne was a terrible loss. Things like that. It was too bad that happened to happen. I want to see what would happen if a life can, what can a life accomplish that has no limit? I want to see that. I mean, the loss of Betty White was a terrible loss of an energy that will never be replaced. I'm tired of that. I don't want to see that. I've been dreaming of this all my life. I've been working on it for 30 years. I don't know how much more it's going to take, but we're doing it. And I'm not giving up. I'm not going anywhere. I'm here to stay. I'm not doing this to make a lot of money. I'm doing this to make the world happier and healthier. And even though the sign says cure aging or die trying, which is my motto, I have no intention of dying trying. But one of the things that people always ask me is, why am I so obsessed with curing aging or preventing this limit on our lifespan? I can't say that telomeres are controlling aging, but this is a scene from Star Trek First Contact. And we all know that someday we're going to make first contact with aliens from outer space. Well, when that happens, I want to be there. And I want all of us to be there. But I think all of us will benefit from finding a way to actually prevent this limit on our lifespan. And to learn more, because I only had 20 minutes today, to learn more about what we're doing, go to www.sierrasci.com. And I want to thank you. Well, thank you very much.
Thank you, Bill. Thank you. That was fantastic. We just have time for one question. Any questions you want to address, for Bill to address? Questions, Moss? One question. Bill, can you hear us well? I'll repeat the question if it's hard for you to listen to.
I could not hear the question.
No, yeah, he hasn't made it yet. Here he comes.
Hi, Bill. Good to see you again.
Hi.
I have a question for you about product B. You didn't mention that product in your presentation. Does product B have an active telomerase ingredient? And if so,
yes.
What's its effect on the telomere?
Well, I typically do not discuss products. I'm a research person, and I get too many cease and desist orders from all the companies that I already sell. I should have mentioned that when I was talking about the supplements and the pet product stuff, there's already several companies that are marketing products that we have discovered, or they contain the ingredients. But product B is now called isogenesis, does contain natural products, nutraceuticals, that do induce the expression of telomerase inside the cells. Okay, that's the main purpose of that product.
Thank you, Bill. Thank you, Moss, for the question. Bill, again, thank you so much for the brilliant talk and for joining us today.
All right. Thank you very much.
Thank you so much, Bill. Thank you. Thank you.